Hello, everyone. Welcome to Feedstuffs 365. I'm Sarah Muirhead. As part of the Horizon 2020 RUMIGEN project, researchers have developed a new chip that stands to revolutionize the possibilities in animal breeding and production. Joining us today are several experts to walk us through those details. Let's start by having each of you go ahead and introduce yourselves and tell us a little bit about your role in the project.
Good morning, Sarah. Thank you for having us today. So I'm Adrian Lopez. I'm a PhD candidate at Eliance in Spain, and I have been developing this EpiChip. So we have been studying methylation marks in the dairy cattle genome.
Yes, hello everyone. My name is Clotilde Patry, and in fact, I'm a doctor in animal genomics. I'm working for Eliance, and Eliance is the French umbrella organization for breeding companies and milk recording organization. And for the RUMIGEN project, I'm in charge of developing the chip, the EpiChip, that is quite new.
So as we walk through our conversation here today, if those of you joining us in the audience have any questions, feel free to post those in the chat area there on your screen. So let's start with a bit about, I know a lot of folks over in Europe know about Horizon 2020, but let's give a refresh here for our US audience and anyone else. And then let's talk about the RUMIGEN project and how that fits in. Who'd like to take that question?
I can try to explain it a little bit. So Horizon 2020 is a competitive cause where the European Union funds research and innovation projects. It has a budget of around 80 billion euros. And RUMIGEN is one of the projects that was funded through this competitive call. So in RUMIGEN, we are trying to define new sustainable and acceptable breeding objectives, taking into account genetics diversity, rare alleles, heritable epigenetics, and the genotype and environment interactions.
So then, Clotilde, what was like one of the outcomes of the RUMIGEN project, and how has that developed into some new technology for cattle, the new EpiChip, I believe it's called, and you can use that to monitor certain traits within cattle. Talk to us about that technology.
I have to say that the RUMIGEN scope is quite broad. We have to acquire data, knowledge, in order to better understand everything around genomics, cattle genomics, so not only the genome, but also the epigenome, and especially in the case of, for example, stress coming from the environment. It could be heat stress, it could be health stress, for example. And in fact, we have worked already on genomic selection, and we will continue to work on genomic selection. And now we want to better understand how the environment can influence the phenotypes of the animals. And that's why we, based, of course, on historical research, historical data, we have tried to develop a cost-effective tool so that we can measure a special mark, which are called the methylation DNA. And we want to measure this level of methylation DNA across the genome in order to first get data, acquire more knowledge, understand how environment can influence phenotypes, and it will be also combined, of course, with the genomic data that we are used to work with for genomic selection. And in fact, the EpiChip, this is one of the output of this project, but not the only one, because as I already mentioned, we are working on several fields. We are also working on genome editing, for example. We are working on societal acceptance, but the chip is part of two work packages that we are called Work Package 6 and Work Package 7. So I'm sorry for these figures, but just to mention that it is just one of the output. And it has been the result of a process. And first, it has been a result of a strong collaboration. Collaboration first in between research partners. Research partners coming from France, Spain, Denmark, that are direct partners of the RUMIGEN project. But we have also used the collaboration and contribution from external partners that are also coming from Scotland, UK, Canada, Japan, New Zealand. And we have accumulated the knowledge on methylation DNA coming from all these partners. Then it has been selected through IT pipelines, and we identified special region of the genome that could be of interest. And we have put everything on a chip that has been provided by Illumina, which is a US company that is also providing the usual genotyping chip. And this is where we are. We have combined everything. We have made some selection from our previous knowledge. And now the chip is available in labs in order to acquire first data.
So lots of involvement from lots of different countries and partners, it sounds like. So then what would you say, what stage of development are you at with this chip? You're still in the research stage, or is it available to ranchers at this time? Where are we at? Clotilde, is that a question that you'd like to take?
Yes, of course. So clearly we are at a research stage. So we just got the chip in the lab and we need to develop everything. So we need to develop the process in the lab for sure, but we first need to go through the technical validation of the chip, then the biological validation of the chip. And based on that, we may for sure evolve this new tool before it can be used directly by farmers.
So Adrian, why is it important for the cattle industry to develop new breeding strategies, to help ruminants adopt to climate change, and what role is this new chip going to play in all of that?
So we know that genomic selection is a cumulative gain we get through new generations. So with climate change, we need to get animals that can quickly adapt to abrupt changes. And this new EpiChip, this epigenetic array, can help us on that. We can select animals that react better to these new changes in the environment without altering their genome. So we can, on one hand, select them by their actual genes, and on the other hand, with this new EpiChip, we can select animals that are more capable of adapting to new scenarios.
So will it evolve? I mean, because climate change is changing, it's constantly changing. Does the chip then evolve to, as that climate change evolves and keeps the industry up to date as to, where it needs to be in some of these things? How adaptable is that technology at this point? Can you say, Clotilde?
What I can say from a very technological point of view is that we can, the chip is composed by marks. And in fact, these different marks can be added or removed. And this is something that theoretically that we can update quite regularly. So we need first to assess the efficiency of this chip right now with the content that we know. We need to accumulate the data in order to associate really what we observe from the chip and what we observe from the animals, the performances of the animals, depending on the environment, depending on the different strains coming from the feed, coming from the temperatures, for example. And based on that, we can make some more conclusion and decide if we want to evolve the content of the chip. And the idea is to get more and more data under different environments. And it's only based on that we will be able to make the chip adaptable. But one of the points is that we already study different traits like health, fertility, mastitis, for example, and other traits, of course, milk production, for example. We need to, for example, have more data in order to include more traits, but we can also test or get data on different breeds. So not only the main large breed, but also other local breeds. And I think it will be a part of the next step also to be sure that the chip can be relevant for different breeds, but also for different environments.
So, Adrian, since both the environment and the animal's own genetics can really kind of create and modify these epigenetic marks and really affect productivity and animal welfare, is this something that's really going to go down the chain and say perhaps even impact future offspring from that animal? Is that going to have an impact through that whole chain?
So there's actually a small debate on the scientific community right now discussing this topic, because it has already been proved that these epigenetic marks can be passed up to 3 generations. So as I said before, it's a tool that could help us to quickly adapt to, because it's not a cumulative gain that we'll get through the generations. But that's something we are looking into, and we don't have much results into that yet. But our hope is that we can use these new marks.
So, in your opinion, Adrian, what's next for this technology?
A lot of validation. We have to do a lot of validation first, and we are running new models to try to include this epigenetic information in the estimation of breeding values. So our idea will be to make it a bit more cost effective, and maybe even include or create a new chip that has SNP positions and methylation biomarkers, so we can combine all this information into one chip, which will be cheaper for the farmers. But by now we have developed some new equations to combine all this information, and it's looking kind of good.
So, same question for you, Clotilde. What's next for this technology? Anything to add?
I think Adrian already told a lot. So about what next, that's a lot of challenges that we have to face anyway. What I can say is that it's very important to make the chip more and more cost effective. So because so far it's a research tool. This research tool will be improved, will be adapted to the different situations and to the knowledge that we will acquire thanks to the RUMIGEN project. And then we will really need to introduce this new tool into the breeding strategies, into the precise farming tools. And the idea is to make a tool that helps the decision of the farmers to adapt depending, for example, on the environment. But for that, we need a tool that is cost effective, that brings sufficient information to take a decision, but that should be also affordable for the different farming situations. So we have still some job to do to make it realistic and to transform this new tool into a tool for the farmers and the day-to-day life of the farmers.
Very good. Any key thoughts you want to leave our audience with today, Clotilde?
I would remind that RUMIGEN is not only this EpiChip, and that we have a lot of other knowledge to acquire in order to adapt to climate change. It's a very, very important challenge for anybody. We have also for sure to take care that any tools that we develop is acceptable by the society, and it should be, of course, sustainable. And so this is something that we keep in mind when we are doing such kind of research. And that's why we think that this research is very important for not only the researchers, the farmers, but also the society. We want to also have a European awareness of all these challenge and tools. And that's why we discuss also this kind of tools within the European Federation, so which is called the EFAB, European Federation for Animal Breeders, where I have the chance to be, in fact, the cattle representative. And it's a good place to discuss research, but also the interest of the farmers or the interest of any organizations that provide services to the farmers. And we would like to use RUMIGEN and any output coming from RUMIGEN in our code of good practice, because at the European level, we have a code of good practice to promote the sustainability of all the tools that serve the farmers and that serve their breeding strategies. So just to leave a message, so we need anyway collaboration in between the farmers, in between the researchers, in between any organization providing services to farmers. And we need to make it available to the European community and to communicate strongly about it in order to promote all these efforts and to be sure that we can continue working on it at the research and practical level.
Very interesting technology, and certainly something we need to monitor and keep moving forward on, because those good practices are always really important to the industry and to consumers and across the board. So I want to thank you both for your time here today. Appreciate the insight and the update.
Thank you very much for having us today.
Thank you.
And we want to thank all of you for joining us as well. If you'd like to watch past issues of Feedstuffs 365, you can go to feedstuffs.com and tune in and watch those on demand and to see upcoming program. For Feedstuffs, I'm Sarah Muirhead.